Hi, this is the Apologist Bookshelf. I'm Gary Zacharias. This book has been called Essential Reading by Michael Behe. He's a professor of biological science at Lehigh University. He wrote Darwin's Black Box, by the way, which was just an amazing book. Talk about an eye-opener. Here's Michael Denton. He's a biochemist. He said, a masterful tour de force, compelling, remarkable. So what are they talking about? Well, there's this book, brand new book out, called Epigenetics and the Architect. And so just briefly, the background of it is, of course, we know that back in 1953, two men, Watson and Crick, discovered the double helix, and that really ignited the whole genetics revolution. But these two authors, their names are Thomas Woodward and James Gills. I'll tell you about them in just a second, but they said there's a second revolution underway. How's that? Well, they've discovered a partner. DNA has a partner in this cellular life. It's called the epigenome. And it's amazing. It's just causing researchers to be flabbergasted. And so this book explores what's going on right now in this unfolding revolution. And, of course, they said, the two authors have said, that we've kind of bought in for a long time that it was all about evolution and that we had random mutations to DNA. And that's how you got to us today. But they said, here's the deal. Much of the information that's used to build biological forms, where is it? It's not in DNA. It's in epigenetic architecture, and that's beyond the reach of random genetic mutations. Now, that's crucial, isn't it? If you can change DNA because of mutation, but you can't change this, how do you get these new body forms? And so these two authors are showing you that it's reinvigorating a whole old field that's being brought out today, and that is that living things look designed. Why? Because they are designed. So who are these two authors? Let's talk about that first, and then I'll get to the first chapter of the book. Thomas Woodward, that's PhD, Thomas Woodward, doctor. He's a professor emeritus at Trinity College in Florida. He's written several books, one called Darwin Strikes Back, which is really good. He's the founder of the C.S. Lewis Society. James Gills, he's a doctor. He's a pioneering ophthalmologist. He's an author of a lot of books. And so these two have really tackled something interesting here. So let's take a look at chapter 1. And they start again back a little bit earlier than today. They said the Human Genome Project got launched in 1990, and the whole point of that was to take the entire human genome down to those exact sequences of DNA's four-letter alphabet, those are chemical letters, that are arrayed along that double helix. So think about the double helix, kind of like a ladder that's been twisted. And if you pulled the ladder apart, you'd have the rungs sticking out. Well, that's the four-character alphabet of chemical letters for DNA. So they started in, let's see if we can chart the entire human genome. That took billions of dollars and something like 10 years or so by thousands of scientists at work. So in 2000, the first milestone was reached. Bill Clinton was president. He called a news conference. He said, we have a rough draft of the human genome. And then they completed it in 2003, and then they published a final draft of that genome. So scientists had delivered a complete map of our DNA. How amazing is that? Right down to the positions and sequences of tens of thousands of genes on the different chromosomes, and each protein-coding gene had all sorts of digital information. And by the way, they had a comment here by Bill Gates, the founder of Microsoft. Does he know computers? Yeah, I would say so. Here's what he said. DNA is like a computer program, but far, far more advanced than any software ever created. Now think about that. Computers run off of a 0 and 1, right? It's a binary digital code. But DNA has four different, what would you call them? Four different bits, numbers, 0, 1, but it'd be 0, 1, 2, 3 if you thought of it that way. So pretty amazing. And so we can now spell out the entire human DNA database. It's over 3 billion letters of DNA. Wow. Well, when people got that, they said, this is great. We're going to have amazing powers of healing. It's going to have an impact. It'll change our lives. It'll change our children's. There'll be new diagnoses, prevention, treatment of all sorts of human diseases. And this was almost like utopia being promised. There's a man who's in the Cancer Genome Project. His name is Mike Stratton. He said, it would surprise me enormously if in 20 years the treatment of cancer had not been transformed. And in Britain, the science minister, Lord David Sainsbury, said, we now have the possibility of achieving all we've ever hoped for from medicine. So that was bright, shiny, look to the future. Wow, things are going to be amazing. Well, as these two authors point out, many of those expectations are unfulfilled today. So they thought maybe they would, when they came out, because they could see where all the genes, where all the information was in the genes, that they would have a lot of powerful gene therapies, but it hasn't happened. And in fact, the New York Times recently had a headline that said, genetic map yields few new cures. Oh, that's sad. And they said that the genetic roots of common diseases like cancer and Alzheimer's says that it's elusive. We haven't found the roots of these. In fact, according to that New York Times article, geneticists are almost back to square one and knowing where to look for the roots of common disease. Isn't that sad? So we did learn a lot by that Human Genome Project. But these two authors said, well, what happened? Why don't we get those medical breakthroughs? Well, one thing they believe was we haven't made a lot of progress in genome-adjacent frontier. We know, starting to know, that there's a second biological encyclopedia of information that resides above the information stored in DNA. So I think that's fascinating. Researchers are decoding another sophisticated software network beyond DNA that's really crucial to figure out how cells develop and how they turn into all the different thousands of cell types during human development. And this new control system, they said, appears to be a factor in aging processes, in cancer, and other diseases. And it seems to guide DNA. It tells different kinds of cells to use different genes and to use them in a precise way that'll meet the needs of these different cells. So what's going on here? This is information beyond DNA. It plays a huge role in each of our 30 trillion cells. It tells the genes when to turn on, when and where and how to be expressed. And that's the epigenome. So they call that biology's mysterious new frontier. Now, they said progress has not slowed. They're coming up with new discoveries, so that's good. But it said scientists are discovering fresh new details about a control system that sits above our genetic riches. So it said much of this architecture resides really close to our genes. It's connected to the double helix in the form of a system of tiny chemical tags and molecular structures. And they call some of these structures nucleosomes. They're tiny spools that are coiled up with DNA. They have a figure here that you can see. You might want to take a look at it sometime. Type in DNA and nucleosomes. That's Nucleo, nucleo, S-O-M-E-S, nucleosomes. So they're little spools that are coiled right alongside DNA. And so they said now it's trying to figure out what's going on there with the genome, and the epigenome is becoming fruitful. And they're saying, again, from a Judeo-Christian perspective, how do you get this sophisticated information processing system to arise if there's only unguided natural processes at work? So they want to define epigenome. So they said it's the various chemical tags that attach to nucleotides, and that directs all sorts of regulatory mechanisms. So I know it sounds pretty complicated, but just keep in mind you have DNA, you have the genes there, but you got other chemical systems that are actually activating and regulating what's going on with DNA. So it's far more complicated than we thought it was. So they said, let's see, here we go. So they said, DNA is flashing a little smile, they said. It's kind of playing hard to get. It's got these scientific secrets. And said, but there's all sorts of helpful clues that are being followed and unraveled. They said one of the clues is the discovery of a splicing code that's inside DNA. So it tells a gene how to hook itself together with other gene products. And the variety of products from one gene, just one gene, what you can get with just one gene is shocking. One person says it's numbering in the hundreds and even thousands. They said it's almost like some chef who's in the kitchen and can produce basic dishes, but can produce recipes that'll produce thousands of different dishes. They said maybe the biggest headline came from something they did a few years back called the ENCODE project, ENCODE. So they said that was an earthquake. So what had happened is that it flipped DNA on its head, how people had thought about DNA. So before they did the ENCODE project, there were all sorts of parts of the human genome that didn't seem to do anything. And they called it junk DNA. So they predicted that probably 90% of our genome, maybe even up to 95%, was composed of just useless gibberish. Why? Because it was debris that kind of accumulated over the years because of evolution. And that's what you'd expect. There was evolution going on. It was fumbling and it had dead ends. And some of that stuff got into our genes there because of mutations, all sorts of random genetic mutations. So you get a lot of garbage, a lot of stuff that didn't matter. Well, along came the ENCODE project. And then they published their findings. And what have they found? So many parts of what they call junk sequences are actually read and copied, and they're used in all sorts of cellular functions. They're not junk. One man, Ewan Burney, he was a pioneer in the field of bioinformatics. How's that for a name? Bioinformatics. I haven't heard of that. Anyway, so he's a pioneer. He estimates that 80% of our DNA does have biological function. He said it's likely 80% will go to 100%. We really don't have any large chunks of redundant DNA. He says that term junk is really not useful. Now remember, before they did the ENCODE project, they thought maybe 90% of the genetic information was just dead-end gibberish. By 2024, the two authors of this book, Woodward and Gills, say 3 scientists completed a review and they found there were over 800 scientific papers describing what was labeled junk DNA and finding out there are functions. They said, of course, we still need to learn a lot, but one thing is already clear. You can toss out the whole idea of junk DNA. They said textbooks are being rewritten to accommodate this surprising reversal. And they're finding out that many of our genes have been found to code for different RNA copies that are never used for protein production. So they said they can regulate our growth, our metabolism. So that's just one thing. That's a miniature RNA, just one of an RNA molecule that's been discovered. They go on inside the nucleus. Pretty amazing. Now, they move along then to the birth of epigenetics. That's DNA's partner, they call it, in the waltz of cellular life. That determines how your genes are expressed. They said, so it's kind of like this. Here's a metaphor. It's not much different from the way a skilled conductor can guide an orchestra. So it's called epigenomics, or more commonly epigenetics. And they have a figure here that compares genetics with epigenetics. So I don't know whether to take the time to do that. Let me do a few of these things. So they compare genetics and epigenetics. So I said key function for genetics is coding for RNA and proteins. What's going on in epigenetics? It controls DNA expression. DNA has a 4-character alphabet, A, T, C, G. And what does epigenetics use? Chemical tags, little chemical tails that are put on DNA, and they're all over the cell. And so said, can DNA be changed by your lifestyle? Can genetics change by lifestyle? No. But can epigenetics change your lifestyle? Yes. And that's the part that's fascinating, I think. So let's move along here. So they said when you zoom in on DNA and you take a look at them, you can see what are called chemical switches. And they said some of them are kind of hard to spot. They're little tiny molecular tags that are on the double helix itself. And sometimes the code is involved in markers that are attached to the spools the DNA is coiled onto. That's called a histone protein. That's getting kind of complicated, I know, but I just want you to get the feeling of other things that are going on here besides DNA. So let me move along here because I don't want to ruin everybody. They said maybe the most sobering discovery from all this research is that if you know what you're doing with epigenetics and you can figure out how to work the system there, it can be modified by our lifestyle. And we can pass that down to our children and grandchildren and maybe even beyond that. So they said it may really change the biology of inheritance. They suggest, recent scientific discoveries say that daily living things like your diet, your stress, your smoking, exercise, if you start living better, you can reprogram your epigenetic system and that of your future offspring. They said, well, how deep do these changes go? How many generations would reap the benefits? They said, kind of fuzzy to know right now, a little hazy. But the point is our epigenome is malleable, and you can make changes that will affect on down the generations. Isn't that wild? That says the age of epigenetics has arrived. Yeah, I guess so. It said it's a multi-layered revolution. So it's genetics, and another layer is epigenetics. Said it may teach us how to modify the epigenome if we'll change our diet, our exercise, and other life patterns to improve our health. So these molecular devices are precisely tailored, and they have these functions that nobody knew about before. Amazing. So I said, we keep talking about it like it's a machine. We said, we're used to machines. But they said, that's only a partial picture. It says, it kind of diminishes in a way the advanced nature of these marvels. They appear to be goal-directed. They can self-assemble. They can self-repair. Our technology doesn't allow us to do that, to self-assemble and self-repair. So I said, can you imagine, take the most advanced human factory that you can think of, but imagine that it could self-replicate, self-repair, and run without any human factory workers. They said, now you'll get a pretty good idea of what's going on with these machines that we talk about in microbiology. And of course, the point as they begin to wrap up the chapter is, so where did all this come from? Was it purposeless change over time or purposeful? So they said, after you look at all this cellular complexity, there's a key question. Who or what designed and built this system? Was it just blind material processes? That's what evolution tells us. Or, and I think this is crucial, is there evidence that all this sophisticated architecture is coming from some designing intelligence, from an architect? And they capitalize the A in architect. And I think that's going to be fascinating. They said in 2022, there's a publication of a new article that came out because people are getting dissatisfied with Darwinism and this whole evolution process because it doesn't explain the kind of stuff going on with DNA. And now with the epigenetics, in 2022, there's an article published in a journal called The Guardian. That's a left-leaning journal. But here's the title, fascinating. Do we need a new theory of evolution? Wow. And so this is science writer Stephen Berani, and he said there are people talking about maybe there's got to be a third way of evolution. They're saying maybe this neo-Darwinian idea that has developed over the years about random mutation, maybe it's just secondary. How do we get new biological form? How do you end up with new information? And of course, you got Michael Behe, wrote a book, Darwin Devolves, and he showed that you need something to create these new genes and these new biological machines. You can't get it from just random mutation. So I think that's interesting stuff. How do you get new genes with all sorts of software-like code? How does that arise in the first place? How do you get this sophisticated epigenetic control system? How do you get that sort of stuff if it's unguided evolution? So as they wrap up the chapter, they say, is it plausible that mindless, undirected natural processes crafted in the cell's hardware and software, can that do that? So it says Darwin himself had no idea. The cell was pretty simple for him back in those days, but now they're calling the cell staggeringly complex, infinitely complex. That's what's starting to pop up in the literature. DNA is so much more information rich than people imagined, and now they got the epigenetic level too. So that's what the book begins with. That's the first chapter. And I realize some of it's kind of difficult to talk about, some of the terminology, when you don't have the book in front of you or have any of the illustrations. But this is not a long book. It's, 140 pages or so. And I'm not going to say it's an easy read because I scratched my head in a few spots. But if you just want to get a feel for what's going on, the immense complexity piled on top of complexity, and to say all of that came about because of random chances, you just throw the dice enough times and you're going to get this. I don't understand how that's possible. Well, anyway, thank you for being part of our podcast here. Hope your day is going well. Talk to you soon.